Good afternoon, everybody. We're going to get started. It's a lovely day. We don't want to spend more time in here than we could be spending outside. So I'm Gwen Beatty. I'm going to be introducing Stefan here today. I think Carolyn is out having a really good time on a canoe somewhere. So our speaker today, Stefan Schmitz Esser, is an associate professor in the animal science department. He's been here at ISU for the last 2.5 years. He came from Vienna, where he was the group leader for the Institute of Milk Hygiene, Milk Technology, and Food Science. And before that, he had gotten his PhD in microbial ecology, also at the University of Vienna. I think it's the same institute, right? Okay. So he has a lot of experience with microbiome in livestock, cattle, but also with the genomics of Listeria monocytogenes. So he's interested in food production systems. And if you look through his recent papers, you see a great diversity of organisms, from pandas to cattle to hard rind cheeses. So very diverse background. So today he's going to be talking about a couple of topics, but the first one will be the contribution of beef cattle GI tract microbes to the tolerance of fescue toxicosis.
Thanks, Gwen, for the introduction, and thanks everybody for coming. And hello everybody again from my side. As Dr. Beatty said, in my group we are interested in many different topics. The kind of largest research area is livestock and livestock microbiota, and this is what I'm going to talk about today. So when preparing the presentation, I originally planned on talking only about beef cattle microbiota and fescue toxicosis. That's would kind of be only as a small or a short time point. And so I'll also talk a little bit about a project also keeping or staying with cattle, and where we have done rumen wall metatranscriptome sequencing today. So first of all, before we get started, I would kind of put up a few definitions, because everybody is talking about the microbiome, the microbiota, the metagenome, the metatranscriptome, and particularly for the first 2 or 3, microbiome, there is no commonly used definition. So some people use it differently, interchangeably, and I will be referring to a relatively recent paper that provided some suggestions how to use that terminology, and that would describe the microbiota as the microbial taxa of a given habitat. The microbiome would be the entire habitat, including the microbiota and biotic and abiotic factors. The metagenome is all genetic material present in samples. So many people use the metagenome when referring to, for example, 16S RNA-based approaches. But from the original definition, the metagenome includes all the DNA. And the metatranscriptome is similar to the metagenome, but here we are talking about the RNA present in an environmental sample. So what we know about the gastrointestinal tract microbiota, particularly if we look at it from a human perspective, that the last, I don't know, 10, 20 years have shown that in humans, the microbiota are responsible for many, many different factors, from normal physiology to a more dysbiotic stage and to disease. So there is these numerous studies have shown that that the microbiota has a tremendous impact on our well-being, our disease, our behavior, nutrition, so many different factors. But if we are thinking about that from a livestock perspective, so what about livestock? And here, the knowledge about the contribution of microbiota to animal production is by far less than with the human microbiota. if we want to analyze microbial communities, I think there are three main kind of research areas that we can be interested. The first is the community structure. Who is there? So that would, for example, be 16S, 18S, or ITS1 amplicon sequencing. The next big kind of building block would be the genetic potential. So what can they do using, for example, metagenome sequencing, genome sequencing, looking at functional genes. And lastly, and I think the most interesting part is to look into gene expression. So what are they actually doing in a given habitat using metatranscriptomics or metaproteomics, for example. And from a livestock, or in my group, what we are interested is kind of all these three kind of factors, and how does the microbiota relate and interact with the animal host, thinking about nutrition, performance, disease, management, stress conditions, and also possibly host genetic factors or breed, maybe things that we are interested in. And I think one of the most exciting developments for being a person working in the microbiome area is the advent of the so-called next generation sequencing technologies approximately 10 years ago. And these are data from the NIH for human genomes, but I think the curves really nicely illustrate how the sequencing costs have dropped during the last 10 years. And this is really unique for us as microbiologists, because this enables us to really analyze microbial communities, composition, and also function in a yet unprecedented way that has not been possible before. So why should we study the impact on microbial communities on animal health and performance? So of course, from a disease perspective that are related to disturbances or shifts in microbial populations. I'll talk a little bit about superacute rumen acidosis later, fescue toxicosis, pathogen challenges. And from a more general production perspective related point of view, so how can we manage to feed livestock in a way that optimizes the diet utilization? And here we can think about, for example, nitrogen efficiency, volatile fatty acid production, which are the key fermentation products. How can we possibly minimize methane production, particularly if we think about ruminants, production performance overall, how efficiently animals can use feed, or can we even maybe use the microbiota to increase livestock resilience? So far for the introduction. In the first part of my presentation, I want to talk about how beef cattle gastrointestinal tract microbiota can possibly contribute to fescue toxicosis tolerance. This is an ongoing project we have in our lab. So fescue toxicosis. Fescue or tall fescue is a grass that is widely used in the US as forage for grazing animals. And this grass harbors an endophytic fungus, epicloe, and this fungus produces alkaloid toxins, and these toxins are toxic for many grazing livestock species, for cattle, sheep, goats, but also horses, and others are susceptible. And this is a huge problem in the livestock production. It causes up to $2 billion of losses annually, because fescue toxicosis can result in reduced weight gains. If we think about beef cattle, for example, lower milk production. If we are thinking about dairy cattle, elevated body temperatures, reduced conception and calving rates. So all these factors are very relevant from a livestock, from a production perspective. And so this is a huge problem. And it's particularly a huge problem in the southern or southeastern US, where tall fescue is widely used as a forage grass, so the so-called fescue belt. And with climate change warming, it is expected that the the tall fescue or the fescue belt, the size will increase and is moving northwards, also into Iowa. And the good and the bad thing about the tall fescue is that this endophyte provides exceptional drought and heat resistance. This is why farmers are using tall fescue. And also the endophyte, the alkaloids, provide resistance against pests such as insects or nematodes. So it's like a, it has a clear good and bad side using the tall fescue. And on the other side, of course, the negative impacts on animal performance. There have been many measures in the industry and the production side to mitigate the effect of fescue toxicosis. For example, by producing endophyte-free fescues, tall fescue strains, which works. But then again, if you get rid of the endophytic fungus, then the heat resistance and the pest resistance gets lost. On the other hand, the performance increases of the livestock increases. People have tried to generate fescue strains that are infected with endophytes that produce lower alkaloid levels or interceding higher levels of different grass, rotational grazing. So there are a couple of measures that people are using to cope with fescue toxicosis. But nobody has yet looked into, or only very limited looked into the microbiology behind that. So our hypothesis is, might the cattle GI tract microbiota be involved in mitigating fescue toxicosis? So this is where we started, and we did some literature research, and there is very limited knowledge about ergot alkaloid degradation, in particular in GI tract bacteria. Some previous studies have suggested that rumen microbial communities are able to degrade ergovaline, so one of those toxins, and other alkaloids, but just on a very general level, so using rumen extract. There has been a link with bacterial tryptophan degradation has been postulated, and a few strains have been isolated, like that Rhodococcus isolate, that are able to degrade ergot alkaloids. So all overall, there's not too much known about the microbiology behind that. So we performed an animal trial at North Carolina State University with Dr. Nick Serrao, which is now in the Department of Animal Science, where 150 Angus cows were exposed to a fescue toxicosis challenge over 13 weeks. So they were fed infected fescue. And out of these 150 animals, 40 were selected based on their growth performance. So out of that 150, we selected the animals who gained most weight and the ones that lost most weight. So the weight gain was our key parameter to select the animals. So we selected the most extreme, so the ones that gained weight in spite of the fescue toxicosis, and the ones that lost most. So the ones that gained weight, we consider the high tolerance animals, and the ones that lost the weight are the low tolerance animals. And we did that at 2 farms, one that was characterized by high or higher fescue toxicosis, and the other one by lower or moderate levels. And so for each farm, we selected again the 10 most extreme animals based on their performance during that challenge. And then we took fecal samples for all those animals and performed 16S rRNA gene and ITS1 region amplicon sequencing to analyze how the microbial communities change in response, or if they change in response to fescue toxicosis. So I'll talk a little bit about the ITS1 region results, which are kind of a construction site until now. So we did sequence analysis. We got plenty of reads and subsample to be able to process them more efficiently, and we ended up with 1,000 OTUs, fungal OTUs. And one thing we were interested in is, are there any significant changes if we look into diversity, species richness, so some more general ecological parameters in those. And farm B is the one with the moderate or lower fescue levels, and the farm U is the one with the higher. And here for the fungi, the results were a little bit inconsistent, so no clear results, and possible increase in species richness in the low tolerant, so the poor performing animals, and some evidence for a decreasing diversity in these communities. If you look at the farm with the higher fescue toxicosis level, there seems to be there's a decrease in species richness and also decrease in diversity of these microbial communities. So this may be a sign of that these communities are more developing in something like dysbiosis state. To look at that from a different level, we performed a couple of community level comparisons. So again, performing the comparing the high tolerance and the low tolerance animals per farm. And these results show that these communities are significantly different from each other in the high tolerance compared to the low tolerance animals at both farms. And if we look at those communities and the changes on a OTU level for the ITS1 region, if you just look at the colors of these of the heat maps, red is high abundant, blue is low abundant. You see there are a lot of different colors here. And what we found is a couple of OTUs. Some of them are highlighted in green. So these are higher, significantly higher in the high tolerant animals. And some of the ones that are highlighted in red are significantly more abundant in the low tolerant animals. So we see distinct changes in these microbial communities. But currently, or what is difficult to assign a possible function to most of these fungal OTUs, because there is just very limited data available. There are some of these fungal OTUs are typical anaerobic fungi, which we find, for example, in the rumen. So this is what we are currently working on, trying to make more sense out of that data. What we see in general is, again, there are OTUs that are more abundant in the high tolerant cattle, and these may contribute to the weight gain we observe in these HT cattle. And what is known that many anaerobic fungi are effective fiber degrading organisms. So many of them can have very beneficial and important effects, and this is particularly known in ruminants. But again, at that time, we are still working on analyzing those data. If we look at the 16S results from that study, again, we did the 16S amplicon sequencing using MySeq, did the sequence analysis, quality control. Most of the reads that we obtained were bacterial. Approximately 1% or less than 1% were archaea, and we found 5,300 OTUs. If we look at the community composition on phylum level, you see there are some significant changes among some unclassified bacteria and among the proteobacteria. Overall, not too much changes on phylum level, but of course phylum is very broad and so doesn't necessarily mean too much. If we look at species richness and diversity estimators for the 16S data, we see a more clear picture compared to the ITS1. Again, these are the two farms and the HT and the LT cattle. And what you see here is then, particularly in the farm U, is a significant decrease in species richness and also in the diversity estimators. And the effect is stronger in the farm U, which has the higher fescue toxicosis level. So also here probably the communities, the fecal communities, evolve or develop into a more into a less diverse state in those low performing animals. Also on community level, we found that particularly at the farm U, the communities are significantly different from each other, and also the statistic values suggest that. And again, with a somewhat stronger effect in the at the farm with the higher fescue levels. Also here, if we look at the microbial communities on an OTU level, again, this is a heat map with red being more highly abundant and blue being low abundant. And so you see, just from looking at the colors, there are quite some different colors showing up between the different farms and the different treatments or the different groups of animals. And also here, similar to the ITS1, we found OTUs which were higher, significantly higher in the HT cattle. These are highlighted in green. And other OTUs which were significantly more abundant in the low T cattle. So there are clear differences in microbial community composition if we expose those animals to fescue challenge. Excuse me. So we try to get a little bit more information. Of course, in general, 16S data are limited because it's just 16S sequences which tells you who is there, but the possible conclusions about the functions are of course somewhat limited. And many of these OTUs that we found are no typical bacteria that we would expect in cattle GI tract microbiota. So if we look at, for example, those OTUs which were significantly more abundant in the LT cattle, we found a couple of different bacillus species, for example, and an Acinetobacter OTU. And so we assume that these may contribute to the low performance of these LT cattle, but at that time we don't know. It's just like kind of correlation or observation based at that time. On the opposite, if we look at OTUs that are higher abundant in the high performing animals, we would assume that those OTUs may be able to alleviate some of the negative impacts of fescue toxicosis and may possibly even degrade those alkaloids. And we try to figure out or learn more about the possible functions of some of these OTUs. Some of these OTUs we found that related species have been shown to degrade cellulose and produce high amounts of volatile fatty acids. And this would be very beneficial for the animal because volatile fatty acids are key to produce milk, to produce meat. And so these OTUs might be able to alleviate the effects by producing volatile fatty acids. So overall, we found a reduced species richness and diversity in the LT cattle. Maybe some signs of a dysbiosis in those cattle. The effects were less pronounced at farm B, which is characterized by the lower ergovaline levels. And both fungal and bacterial OTUs changed in abundance. And there are ones that are higher either in the LT or HT cattle. So obviously what we can say is that fescue toxicosis does induce significant changes in fecal microbial communities, and particularly if we compare those, the animals based on their performance. So we assume that some of those microbes are involved in mitigating the effect of fescue toxicosis, but of course the big question is how, and we can't answer that right now. What we want to do here, and I go back to my introductory slide, is what we want to do is to do metagenome shotgun sequencing to learn more about possible metabolic pathways or functions that may be more abundant in those HT cattle compared to the LT cattle microbiota, to be able to tell more about why these microbes are changing in abundance. In the rest of my presentation, I want to focus on another project that we have in cattle, where we use metatranscriptome sequencing of rumen wall microbial communities. And first of all, I want to give a very brief introduction into the rumen and into rumen microbial communities. The rumen as the characteristic fermentation chamber of ruminants has 2 or 3 different, largely different microbial communities. So there are microbes in the rumen content which are attached to particles or maybe a part of the liquid fraction. And there are bacteria on the rumen wall, and we are interested in the latter ones. The entire rumen, or almost the entire rumen, is covered by finger-like projections, so-called papillae, and they can be fairly large. And these are increasing the surface. And the rumen wall is important for absorption of fermentation products, such as volatile fatty acids, but also, of course, the rumen wall is the interface between the host animal and the rumen content with the microbes. So it is a very important barrier also from a health and performance perspective, because what many studies have shown that under stress conditions like heat stress or low pH, inflammation caused by LPS, many animals develop something called a leaky gut, so where the integrity of the epithelial or mucosal tissues is impaired, and this has very bad impacts on animals' health and performance. And again, the rumen wall, what is known, it is, as it is the barrier between the rumen content and the host animals, there's a lot of nutrient exchange going on at the rumen wall. And what has been known for decades is that almost the entire rumen wall is covered by bacteria. So these are some scanning electron micrographs of rumen wall tissue, showing that all the different microbes and different morphology of the microbes that cover the rumen wall. And here are some fluorescent, this is a fluorescence in situ hybridization image where you can see the microbes covering the rumen content in red. The rumen content, the rumen wall in red, and also this is a histology stain, and you can hopefully see the microbes here. So the rumen wall is covered with microbes, and many studies have characterized the composition and those rumen wall microbes. So we have a decent knowledge about who is there, but our knowledge about what they can do and about the function is really very limited. So what we've done back when I was back in Vienna is perform an animal trial where we induced so-called superacute rumen acidosis, SARA, in dairy cattle. And this is a very common metabolic disorder in cattle that can be induced, for example, by high energy diets or rapid increase in grain feeding. This leads to a decrease in ruminal pH. And then this can have bad impact on animal performance. So we had an animal trial with 8 Holstein dairy cows, which were fistulated so that we can access the rumen content and take samples. Here you see the rumen wall and the fistula. So we induced SARA by increasing the grain content in the diet, and we took samples at baseline after 1 week of adaptation and after 4 weeks of the high grain feeding, and performed as a first step 16S RNA gene-based MySeq sequencing. And what we could show, and you don't need to go any kind of in too much detail here, if you look at the heat map, the different colors, again, different colors mean different abundance. So, and also if you look at the PCOA plot, so you see those rumen wall communities change significantly in their composition in response to that SARA challenge. Again, as I said, the function about those rumen wall bacteria is still very low. So we have no idea. So we know they change, but we don't know why do they change. And so to analyze that, we performed metatranscriptome sequencing of those rumen wall biopsy samples to analyze what is the actual gene expression of the microbes under these conditions, to learn more about the function of these bacteria. This is, so what we did is take rumen wall biopsy samples and did RNA extraction and depleted host and bacteria RNAs, did Illumina high-seq sequencing, generated many reads, and analyzed that those reads to see and to learn more about the function of these bacteria. If we look at the taxonomy of these bacteria reads, so I'm only talking about the bacteria reads now, you see the high abundant ones in blue, and the red is low abundance. And so you see some of the phyla that were abundant in the 16S data also here. And so classic phyla being abundant proteobacteria, Firmicutes, Bacteroidetes. So this is pretty typical. If we look at genus level, we also found a couple of genera to be abundant on the RNA, on the metatranscriptome level, that we also found to be abundant in the 16S-based DNA studies before, like Campylobacter, Bacteroides, and Neisseria, and others. So we can use the RNA-based metatranscriptome data to learn something about the taxonomy of those microbial communities. But the most interesting part in our perspective is not so much the taxonomy, but the function. So what genes are actually expressed. And this heat map and the table shows the 50 most abundant bacterial hits. And I don't know whether you can see it. Most of that, if you look, is general, like housekeeping energy, ribosome, glycolysis, pyruvate, metabolism. So from a more general perspective, not too exciting, but still we learned something about the function. And of course, general in gene expression studies, most of the housekeeping genes are of course the most highly expressed most of the time. But as we have that data, and we can mine and search those data for genes or pathways that we are interested in to learn more about. And I just want to give a few brief examples here. Some early studies back in the 1970s have suggested that one function of the rumen wall microbes is urea degradation, so that they have urease activity. And this has been confirmed in a more recent DNA-based study. So that's why we decided, okay, let's look at nitrogen metabolism genes. And so you can again mine those data for genes involved in nitrogen metabolism. And what we could show, for example, is that indeed the urease is among the most highest expressed genes of the microbial community. So indeed they are actually degrading urea at the rumen wall under those conditions. And then we can oftentimes break it down at least to genus level, so which organisms specifically have that capability. And we also found other key enzymes of the nitrogen metabolism highly expressed in the rumen wall microbial communities. And we also found evidence for nitrogen fixation happening at the rumen wall. This is something that has been shown and speculated earlier. So, and again, for certain, not for all of the genes, you can break it down at least to the genus, so you can tell, okay, this is the activity, this is what the gene expression is, and which organisms are performing that activity. We looked at other metabolic pathways. One important is starch degradation, and also here we found high level of different starch degradation genes, for example, the alpha amylase. And again, we can tell which genera or which bacteria are responsible for that. And also evidence for degradation of cellulose and cellobiose. So really we can mine those data to look for genes of interest or pathways that we are interested in. Of course, if we are thinking about mucosa or epithelial surfaces, short-chain fatty acids like butyrate or propionate are key for intestinal health. And what we could show is that two pathways for butyrate production are expressed by the rumen wall microbes. And again, we can break it down to the genus level, and also one pathway for propionate production. So most likely the rumen wall bacteria are producing butyrate and propionate actively on the rumen wall. Somewhat surprisingly, we found high levels of gene expression of genes involved in oxidative stress response, which is somewhat surprising because the rumen is per se a strictly anaerobic ecosystem, but many of these genes were highly expressed, or among the highest expressed genes in different organisms. So we think that maybe a response to diffusion of oxygen from the host tissue, because the host tissue, of course, is an aerobic ecosystem. And so we think that this is probably why they express those genes to cope with this. You know, if these cells are living in a biofilm directly on the host tissue, there is diffusion of oxygen. So we think that's a response here. So overall, with that study, we could show or could reveal first insights into the function of rumen wall microbiota. We show that housekeeping genes, not surprisingly, are among the most highest expressed genes. Nitrogen metabolism seems to be a key function of those rumen wall bacteria, particularly urease activity. They are actively degrading starch and cellobiose, producing butyrate and propionate, and they are actively involved in oxidative stress response. When we look at the main function, or one of our other main questions in the beginning, when we looked into the baseline and SERA, so stress conditions, and we didn't find a lot of statistically significant differences. In our metatranscriptome data, so this may be due to a low number of animals that we've used for the metatranscriptome sequencing. Sequencing depth may not be sufficient, or maybe the mapping of the reads to the reference database may be suboptimal, because what we know is that these, from 16S studies, that many rumen wall bacteria show only very low similarity to characterized and culture type strains. So it may be that at least some of those communities are simply too different to be kind of covered, or that we can map them efficiently, for example, to KEGG or other reference databases. And because we did, and I mentioned that we did take rumen wall, so we did take these tissue biopsies. So, and I talked only about bacterial gene expression, but with our RNA extraction and the RNA depletion, we did sequence both microbial and host tissue, so bovine gene RNA. And so what we did is bioinformatically separate the bovine reads from the microbial reads. But of course, analyzing the gene expression of the host tissue could also be very interesting, because then we can possibly link the gene expression of the microbes with those of the host. And this is something we just started with, looking a little bit into gene expression of the host tissue. So we find under SERA condition a downregulation of genes involved in tight or gap junction, desmosome, cytoskeleton, which is kind of makes sense, because under SERA condition we see things like a leaky gut, so tight junctions get more loose or not so tight anymore. And so we see upregulation of genes involved in epithelial differentiation, genes involved in immunity, and urea transporters. And also the urea transporters make sense, because the urea is transported from the host tissue into the rumen, and then the rumen wall microbes are sitting there and degrading the urea, So these are just some very first insights into some of these gene expression patterns, and this is at that time clearly really more the tip of the iceberg here. And as a microbiologist, trying to make sense of eukaryotic gene expression is can be pretty challenging because microbes are complex and eukaryotes are even more complex. What we'll do with those data, and I talked about, analyze bovine tissue gene expression in more detail. And what we are working on also is to apply maybe a de novo metatranscriptome assembly and or different reference or mapping approaches to try to learn more about the function of these rumen wall microbes. And with that, I want to finish and thank particularly Luke, who did the work on the fescue, and Nick from our department, who worked on the fescue, and some members of my former group back in Vienna. And I'd like to thank you for your attention, and I'll be happy to take questions if there are any. Yes.
In the first part of your talk, when you have OTUs,
yeah,
you were able to say that you think some of these are volatile fatty acids.
Yeah.
I'm guessing that's based on the predicted genus or phylum or whatever.
Yes, and what related species, this is of course based on 16S, so this is pure kind of correlation only. So because the related organism can, produce volatile fatty acids, we assume that those OTUs that we actually found do the same. Of course, this is no proof by any means.
It just made me wonder how, what you would predict for the variability in volatile fatty acid production. Most organisms in one genus that make it produce it, or is that something that depends on genes that are likely to vary strain by strain? And the cross bacteria, is this a really common function?
It's not that common. It's not that common. So there are a couple of studies that have analyzed it in the human and other microbiota. So it seems to be, maybe something like species or genus level like function. If you look at key functional genes for butyrate or propionate production genes, but of course this can also be very strain dependent. And so this is of course one of the key limitations, that if we work with 16S data and try to, infer potential functions based on just the 16S as data. So this is why we clearly need some functional data here. Yes.
So, on the first of the studies that you presented, were all the samples were taken after the treatment diets were applied?
Yes, so they were fed the fescue infected diet for 13 weeks continuously, and then we followed their performance, the weight gain, and at the end of the trial, all were sampled again after 13 weeks, and then based on the performance, weight gain or weight loss, we selected the most extreme animals in either direction.
So in that case, you don't have an idea of which of the more tolerant or less tolerant cattle actually might look more like?
Yes, so we don't have a classic negative control, like in a way of an untreated or controlled treatment. Here the idea was really, and this has been proven pretty useful in other studies that looked into feed efficiency and other traits, that if you select the most extremes, you can try to, or the hope is that you see clearer effects. But yes, in that case, we did not have a kind of classic control in a way of no treatment at all. That was really the reason or the criterion to select samples was the performance, the weight gain. Yes?
In the cattle population you were using, would you be able to do genetics on the cattle side to understand what might be driving the microbe populations, or?
That, I just realized I forgot to repeat the questions. So the question was that whether we had looked, or we're looking at the host genetics. Yeah. That is something that Nick Serrao is looking at into. currently. So that's the other potential side we want to look at, and that's very well possible as well. So the host genetics may, of course, be a factor that contribute to that. And yeah, in that study were only interested in the microbes, yes. And so that may very well be, but I have no data on that. Yes, please.
And more generally, in doing metatranscriptomic studies, you in the end say it's hard to see treatment differences.
Yeah.
I'm wondering if working with a statistician, if you could predict how many samples would you need to see differences, or is it kind of hopeless?
Well, I think in that particular case, I don't think it's hopeless. I think the main limitation in our case is that the low number of samples for each group. And, you know, there is, of course, a high variability. So this is certainly something that will contribute to that. And we've been very stringent with our gene expression, like the criteria, what is the differentially expressed genes, but I think there is no other way to do it. So this may be one factor that contributes, and we think that particularly because we know from 16S studies that they are so different, we think we'll lose a lot of reads because we can't map them to the KEGG reference database, because these communities are so different. That's why we are trying a different approach with the same data set to do a de novo assembly so that we get longer reads, and so to try to see whether, because yeah, I don't know, I don't think it's not hopeless. But it's, of course, particularly with these tissue samples, the big challenge is, of course, a lot of the vast majority of host reads that we sequence, and even with the new high-seq sequencing technologies, you need to do a lot of sequencing. And it's feasible, and it's exciting, but it's, of course, another challenge that there's such a lot of. And of course, if you think of a little like papillae, the most of it is, of course, host tissue, and the host genome is, of course, 3 billion base pairs and not 3 million base pairs, like an average bacterial genome. So there are a couple of limitations, but also, I think, many opportunities. Yes, please.
Do you have plans to look at rumen microbiota with regard to the fescue toxicosis?
The question, and I apologize for forgetting not to repeat the question. The question was whether we were planning to look at rumen microbial communities for the fescue. Yes, indeed, that's something we want to do. So in that case, because that was a pretty big trial, we took fecal samples because it's the most easily accessible sample material. But yes, that's something for the we definitely would want to look into, is to sample rumen content samples and to see what is actually going on here. But based on the results from the 16S analysis, we are pretty confident that the microbial community does contribute to that, and that it's worth analyzing in more detail. James.
With the subacute ruminal acidosis study, have you looked at varying levels of pH and how it may impact the actual physiology of the animal, the papillae?
So the question was whether we looked into varying pH levels for our metatranscriptome data, for the SARA data. We had only 8 animals, and what is known with SARA is that that different animals respond differently. So there are, like, if you just look, some cows will develop a superacute rumen acidosis and others will not. So there are responders and kind of non-responders from the animal side. And in that case, we didn't look into all of those cows had a pH below 5.8 or so for at least, I think, 5 hours a day. So that would be considered as SARA conditions, but they were all, the cows that we used, they were all similar in the drop of their pH. But there are definitely differences between different individuals. So there is a lot of many more things that we can analyze here. Absolutely. Are there any additional questions?
Maybe online.
I can't see anything here. I've got the window here.
If
Yes,
you think
please.
on your last study where you sampled the rumen.
Yeah.
If you took fecal samples at the same time, do you think that would allow you to add or subtract groups of bacteria that were active or not active? Do you think that's completely a different story?
So the question was whether if we would have sampled fecal sample in addition to the rumen wall biopsies, whether this would be, maybe may allow us to better differentiate active from non-active communities. We did take fecal samples and also rumen content samples from the same time points, and both of them are just from 16S-based community composition pretty different, and particularly the rumen wall microbial communities from the rumen content. And then if we compare those to the fecal communities, this is kind of even more different. So I guess that's probably wouldn't help us too much, I guess, and because they are, well, of course, fecal samples are easy to get, but of course it's the end of the GI tract. And so it is different. It is definitely different.
If there's no more questions, I can join you in thanking everyone.